that comes and releases, and it opens up these positive ion channels. Remember, I talked in another lecture about acetylcholine. Acetylcholine will bind to these channels and it allows sodium to come inside. Then it'll make this positive and that'll spike, and that will cause a reaction. Opens up those channels, and then we start to get an electrical impulse. And so that's where I'm going here. So that stimuli will affect the brain, affect the membrane potential. It allows sodium in, and then we get this positive influx, or this big increase. And so that starts the electrical potential when the sodium ions plugged in, and they change that, and that stimulus goes, and that stimulus then moves down the axon. And as it continues to move down the axon, it still opens up, allowing those in, and basically goes from a negative to a positive, and that's what depolarizes and comes down the cell. And it continues to move down, and that action potential keeps being propagated down the entire way until it ends, or we stop what it needs to do, or it completes its function from there. Skeletal muscle fibers have axons that are excited, and that basically connects it. We don't see it so much in the smooth. The smooth muscle has its own reaction on how it works. So really, we're talking about these action potentials. We're talking about the skeletal muscle fibers as our main components that we're doing. And I'm going to skip through some of these slides here. I will get you all these slides as well, but I want to talk about some specific ones and go through, because I'll mention from here. So again, we have our nerve, our axon coming down. Our resting potential is a negative 70 millivolts. Once we get a action potential that comes down, it comes down here, it opens up this threshold, allows sodium inside. This influx of sodium causes that area to depolarize, or we get that motion to come out. Once it comes out and we get the action that we needed, then it closes up all those channels, starts pumping out the sodium and potassium back out of the air, trying to get down to negative, and it starts to repolarize itself, getting ready for the next impulse. Comes back down into this refractory period, and then once we get balanced, we're back down to that resting of minus 70. And then the nerve is ready to send another action potential. Now this happens really fast, multiple times, so the body is able to move nerve senses very quickly. So, and then we're looking for that action potential to reach the area we go. So again, here when we're talking about this nerve, as we're traveling down, we get the stimulus, sodium comes in, here's where it's still balanced. It'll slowly come down, and the sodium will start coming into the area, and as it moves down, it comes down and it causes that depolarization, and it keeps moving down this whole axon. As it's doing that, this part starts repolarizing. Here, I apologize, starts repolarizing. It starts pumping out those sodium potassiums back in to get that action potential back down. So this moves that current down the axon. Now when we have myelinated areas on here, it will jump very quickly, because then it doesn't have to cause the action potential just in those nodes. And that's what this myelinated fiber is going to talk about. So again, we're dealing Schwann cells do the myelination in the peripheral nervous system, oligodendrocytes in the central nervous system. And here's that area now. So if we have the myelinated sheath, you can see it only has to depolarize here, then depolarize here, then depolarize here. And you can see that quick motion, so it can repolarize it, and that allows that action potential to move so much faster in a myelinated sheath versus a non-myelinated sheath. Neurotransmitters is basically what allows us to take that nerve impulse and get that information over to either another neuron or the effector cell, whether that be a skeletal muscle, whether that be another type of receptor, whatever it is. So we have our synaptic cleft, which is basically the space between the synapse of the terminal section of the nerve and the area that we want it to work on. In that synaptic terminal, we have some parts here. This is where I'm going to focus on this electron microscope of what this kind of looks like. But here, what we have is we have mitochondria in the synaptic area. We have all our synaptic vessels, and these are what carries our neurotransmitter, which we're going to talk about mostly acetylcholine. Here's the synaptic cleft, or the space in between, and this has got to be very close so these transmitters can go right over here. And then the postsynaptic membrane, or the membrane we're going to act upon, has little receptors that those neurotransmitters, when binds to, is going to allow the influx of sodium, potassium, whatever the areas it's going to need to transfer that nerve impulse. So that's really what we're looking at here, and I really like pictures for that. Acetylcholine is going to be our main one that we're talking about. There's other type of neurotransmitters, but that's the main one we're going to focus on as well. And it will basically be in these synaptic terminals. It will cross the membrane and it'll bind to these receptors. As it does, it releases the acetylcholine here with that nerve impulse, and it's going to start to bind here to these sodium channels to allow sodium to come in. Once it has bind, the sodium influx into the cells, and then that causes the reaction that we want to do. And then once we're done, acetylcholine esterase will bind with the acetylcholine, has a high affinity, because now we've stopped our nerve impulses, so everything that's left in here, it starts binding to and collapsing it, so it closes off the sodium pump, and then sodium channel, and then the sodium starts getting pumped back out to that normal resting potential. So that's the function we're just seeing at the neurotransmitter section. On the skeletal side, we already talked about it a little bit more in depth. Other neurotransmitters we'll be dealing with, norepinephrine is another one, or noradrenaline, dopamine, GABA, serotonin, all different type of neurotransmitters that we'll see. Again, our main one we're going to be dealing with, though, is acetylcholine. Why this is important, too, is there's acetylcholine esterase, which stops the nerve transmission. Well, what do you think we have that's similar to acetylcholine esterase? Well, we have succinylcholinease, which also is a competitive inhibitor of acetylcholine and stops that neurotransmission, so we can basically help paralyze. So we'll talk about that's where these areas function at when we're using medications. And we'll talk about these synaptic areas, and I'm not getting too much into it. Again, we're going to get more into these into the actual class, but that's basically what I really want you to know on the main aspect. So the other thing we have is we have these neuronal pools, or basically it's one nerve. Sometimes it's got to act on several nerves. We've got one impulse, but we have to act on several things. I want to move my fingers to hold a pencil. Well, I'm going to act on all these. One impulse is going to come down and act on multiple different ones, because I've got to get my index finger, my middle finger, my thumb. Everything's got to be in coordination, so I'm going to work on multiple different nerves. So I got to get these sensation downs. That's divergence. I mean, it's diverting into multiple pathways. Opposite of this is convergence. Basically, we need one action that's going to be done, but we need multiple different aspects that's going to go on here. So we need to work on multiple different aspects of this to get this come in and get multiple area. And this comes more with breathing and everything like that we're going to be doing and getting it to hold. So all different functions that we're going to go. But again, this is a convergent pathway where a bunch of nerves are converting onto one nerve and getting the one function that we need to do. All right, and then the central nervous system is going to be our main area. This is going to be everything from our brain to our spinal cord. Because this is very sensitive to a lot of different things, it needs to be protected. And how we protect it is through the meninges layers. The meningeal layers are 3 layers that help cover and help take care of that. They're specialized membranes, and they each have their own function on here. So let's talk about each of those. So our main 3 layers, the dura mater, we have the arachnoid, and the pia mater. Well, the dura mater is basically the hard surface layer, and that's going to be what's doing the most protection. So it's the outermost layer. It's right usually attached to the cranium or the skull, and it basically is the most protective. It's the thickest of the layers. The arachnoid area basically is the space in between, and it has, as you can see, kind of these spider web cells, and the the cerebral spinal fluid floats inside this arachnoid space, and that helps protect there. And then the pia mater is the most inner layer, and it's what covers the spinal cord and the brain specifically. The pia mater will form to the gyra up into the brain, into all the holes, and it'll wrap perfectly right on the edge to that, and it helps protect that way. So again, the dura mater, the most outer layer, fibrous layer, it's the toughest layer. Spinal cord, the dura mater again is the most outer layer, and it covers everything up and goes down to the epidural space, which we'll talk about here in a little bit. The arachnoid is the inner layer, and it's a large number of squamous cells. It has a space into it in between the dura mater and the pia mater, and it basically allows the cerebral spinal fluid to bathe that area and contains that area. The cerebrospinal fluid is a very thick fluid. It's very viscous, but it has all the nutrients and chemicals, ATP, sugar, all this products that comes in, as well as getting rid of the waste. It's like that helps bathe and take care of all the nerve tissues. Besides just the blood flow, this is what else helps take care of that. And then the pia mater, as we talked about, is the most inner layer. It's the closest to the nerve tissue and the blood vessels in the brain and the spinal cord, and it's the most inner layer that wraps specifically to the spinal cord and the brain. Highly vascular, and it's, again, very supportive on there. Then we have the spinal cord, which basically is the communication factor of the brain down the spinal cord, and then it breaks off into the central nervous system. This goes through the foramen magnum out of the skull when it comes down through the spinal canal, and then peripheral nerve roots jump off this and go out to the body. Don't need to know too many specifics here. I'm going to try and just keep you on the information you do need to know and skip here. But here's a good look of that spinal cord, a close-up look here. And what we're going to see is that spinal cord will come right down, comes all the way down. The spinal cord itself ends at about between L1 and L2 is the end of the spinal cord, and then we have what's called the cauda equina, which comes all the way down and has little nerve roots coming out. We call it the horse's tail because now this is just nerve roots. It's not this full spinal column that looks like this. And in the spinal column, again, the gray matter is all our nerve cells. The white matter is all the axons, mostly the myelinated axons around. So we have a dorsal root and we have a ventral root that come out of this. Again, the dorsal, when we're referring to here, is the posterior aspect or along the back. The ventral is along the stomach side or the front. And that goes down the canal. And then we have these nerve roots that come out of here. So out of this area here, we have the posterior horn and the dorsal root, and then we have this larger ventral horn and the ventral root that comes out here. So this portion sends out the motor from the ventral aspect, and then we get sensory coming in here as well. The spinal cord consists of 31 segments. Again, we're talking about all the roots come out below. There's one set above, but we're coming below each of the spinal columns. So again, I want you to know all the areas: the cervical, the thoracic, lumbar, and sacral, and how many bones are in each of those sections. And then the nerve roots come out of each of those areas, and then that's how we name their roots. We have the dorsal roots, which contain the axons of the sensory going out, and the ventral roots, which have the motor nerves. And then the intervertebral foramen is the space in between there. So spinal nerves, these are what comes outside the vertebral column and come out, and they'll meet up with forming the peripheral nervous system. And then the cauda equina, again, is all these nerve roots that come out the tip of the spinal cord. Looks like a horse's tail because it's all a bunch of just nerves coming down. Here's another look at that. Again, ventral, dorsal, and our functions that are going to be coming through each. Discussed the white matter already, and then gray matter.